Okay, we've developed a new design for a protocell for a protoorganism, which is based on a minimalistic approach where we make sure that the three important components that contribute that are necessary for making a functional protocell, namely a container and a metabolism and some kind of protogenetic machinery, thermodynamically will assemble. And we do that by starting out with a micelle, which is a small structure consisting of surfactants or simple lipid molecules. We then in this micelle assemble, or rather we put into this micelle peptide nucleic acid, which is different from DNA or RNA because it has a peptide on the backbone, but else it looks the same, it has the same kind of basis. And due to the hydrophobic backbone, it will stick to this lipid aggregate. So now you have the lipid aggregate and the proteogene together. You then need to supply a photosynthesizer molecule, a molecule that's able to capture light energy, and these molecules are cyclic hydrocarbons, so they will also stick to this aggregate. So you now have all three components together that's necessary for this little thing to self-reproduce. And the way the life cycle goes like this: that you have you feed the system with precursor molecules, which consist of molecules that, after they are broken up, can form new lipids. So they act a little bit like hydrocarbons or oil. So this aggregate initially it will swell up, while all this hydrophobic stuff sits inside it, and then as you shine, and that's one precursor molecule. The other precursor molecule is, say, two complementary pieces for the PNA, the protogene, and if you make sure that these things can't ligate before they have gone through a particular process, then they will also qualify as part of the metabolic system. So you now have everything with all the precursor molecules ready, and then you shine light on it. And what happens is that you break, with a photofragmentation reaction, you break these precursor lipids up, so you form lipids and some waste. And what will happen is that this aggregate will become unstable because you'll form more and more of the lipid molecules. They'll break up, and this has to be coordinated with. As that happens, we need to also ensure, and that will happen thermodynamically, also by the PNA template will take the two complementary pieces and align them, and then as we shine light on the ends of the PNA, we'll break the pieces off that are spurious, and now we can make the peptide bond, the amide bond, and experimentally we know that this is possible to make amide bonds. Luigi has done that. Actually, a number of people have made these experiments in different contexts. So the key issue is, of course, to put them all together. So now you have two PNA molecules, and then I think the most difficult part of this system that we have not tested yet is to make sure that you design the system in such a way that it will dehybridize, so that once you have made a new template, they will not stick so strongly together that they can't be separated when you have this lipid aggregate to separate. Now the real interesting part is that without the PNA and a particular sequence of this PNA, this photofragmentation reaction doesn't work very well. The quantum yield is only 2%. Whereas if you have a guanine and an adenine sequence, then they can work as an electron relay system. So that means that you have you actually have a coupling physicochemical coupling and thermodynamical coupling between all elements, namely the protogenes, the container, and the metabolism. So you can actually do evolution because the genes with GA, the GA genes, together with the aggregate, they will be able to generate much more new stuff, new building materials, so they will perpetuate, whereas another poor protoorganism that doesn't have the GA in the DNA won't be able to do that. So that's sort of the key. That's I don't know, 2 or 4 minute story of our protoorganism.